So, good afternoon, guys. And today we'll be starting with the second class of our discussion week 6, and we'll be discussing about the leftover previous year questions and further moving to the eukaryotic transcription. Okay. So, in the last class we have discussed about the tryptophan operon, bacteriophage lambda transcriptional switch, and some PYQs related to the bacterial transcription. Now we'll be moving on to the previous year questions related to the lac operon and the operon system. Okay, so this was the question that was recently asked in GATE 2024 BT. Okay, so which of the following conditions induce the expression of beta galactosidase gene in the lac operon? Okay, so yes, as you guys might know, that Kate also have uh, two kinds of questions. One is single select or single answer questions, and one is multiple select questions. Okay, so in this case, we have multiple options that can be correct. Okay, so just keep this in mind while answering this question. So, which of the following conditions induce the expression of beta galactosidase gene in the lacoperon? Okay, so the options are absence of glucose, absence of lactose, presence of glucose, or presence of lactose. So, first we'll draw the lacoperon. Okay, so this is the promoter, the operator, then we have the Z, Y, and AG. Okay, so this is the basic operator. This is the basic operon, lac operon. Now, what we'll have is if the lactose is absent. Okay, so the repressor will bind to the operator. This is the repressor, and the RNA polymerase cannot move, so there is no transcription of beta galactosidase. So, for the expression of beta galactosidase, lactose must be present. Okay, so this was the first thing. Now the second thing is the based upon the utilization of different sugars at that point of time. So we discussed about the presence of glucose and lactose in the same medium. So if both of them will be present, then the expression will not be that proper of these particular structural genes. So what will happen? Very less expression will be there. But if glucose will be absent, so here we'll have two conditions: glucose present and glucose absent. So in this case, there will be very limited expression that will be considered null, because lactose was present, so some leaky expression would be observed. But if glucose will not be present, so in that case, there will be proper expression of beta galactosidase because of the CAMP proteins that favors the transcription of the structural gene of the lacoperon. Okay, so the beta galactosidase expression would be visible only when there will be absence of glucose plus there would be presence of glucose. Okay, so we have fourth option, that is the presence of glucose, presence of lactose, and when there is absence of glucose. So the correct answer is option 1 and 4. So this was a MSQ type question. Okay, now moving on to the next question. With reference to the lac operon, what will be the phenotype of the E. coli strain having a genotype I minus, O plus, Z plus, Y minus, slash, this is the F prime. So this part is present on the plasmid. We have I plus, O constitutive, Z minus, and Y plus. Okay, so what will be the phenotype of this particular strain. So what will happen given this phenotype? So remember we discussed about different mutations that are present in the lac operon. So as per the phenotype, this is the genetic material wala part, and this will be the plasmid present on the plasmid. Okay, that's why has been given as a diploid chromosome. So considering the genetic material wala part first, so the genotype is Y minus, and for the plasmid one we have I plus, OC, Z minus, and Y plus. So from the genetic material wala part, this will not be produced. So in this case, the repressor is absent. Okay, so repression is mutated. Operator is normal, so that it will be bind. It can bind to the repressor, and then we have beta galactosidase normal, and lac permease absent. Okay, now we'll see the genotype of the plasmid wala part. So in this repressor present, then we have the OC, that is constitutive operator. Okay, and then we have beta galactosidase absent and lac permease present. So now how will we use this information to deduce the answer? Okay, so what will happen, as in the here I'll draw so that will be easy to understand. So what will happen here, we have the promoter, operator, Z, and Y. Okay, so the repressor is absent here. Okay, so considering this as the repressor, this is not present, and this is positive, this is negative. Okay, so no lac permease will be present from this case, but what will happen, we have the repressor from this particular plasmid protein is working fine. Okay, so it can go and bind to this operator. Fine. So what will this do? It, because it is a transacting sequence, so it will bind to any sequence related to the operator where it is present. So this particular repressor of the plasmid can bind to the operator of the genetic material also. Okay, so what will happen, it will bind and it will not be able to produce the beta galactosidase. It will only produce the beta galactosidase only when the lactose or allolactose is present. Okay, so the production of beta galactosidase here will be inducible. Now, talking about the plasmid gene, uh, here it's an constitutive operator. That means the repressor cannot bind to this particular operator. Okay, it cannot bind here. So it will not be able to hamper the production of any of these genes. And here the beta galactosidase is absent. So here no production of beta galactosidase. So the production of beta galactosidase will be inducible, and then we have lac permease. We'll see about the lac permease. So in the genetic material wala part, there is no lac permease, so no question about the lac permease. Now because here it's a constitutive operator and here the repressor cannot bind in the plasmid genome, so here the lac permease that will be produced will be constitutive. Okay, because there is no hamper or no regulation in its production, so it will be constituted. Now we'll see what are the options that fall correct in this. So we have constitutive for both beta galactosidase and lac permease. No, this is not the case. Then we have inducible for both beta galactosidase and lac permease. This is also not the case. Then we have inducible for beta galactosidase and constitutive for lac permease. Yeah, this is the answer. So the correct answer is option 3. So yes, the correct answer is option 3. Okay. Now moving on to the next question. The lac operon in E. coli is controlled by both lac repressor as well as the catabolite activation protein CAP. In an in vivo experiment with lac operon, the following observations were made. So we have CMP levels high, then repressor is bound with the allolactose. Okay, so repressor has already been formed with the allolactose. Then we have CAP interacting with the RNA polymerase. Okay, CAP also interacted with the RNA polymerase because CMP concentration was high. So, which of the following conclusions is most appropriate based on the above observations? Okay, so based on these observations, what we can deduce is there will be the lacoperon is basically active. Okay, why? Because this is the CRP CMP binding site. This is the operator, and then we have the promoter. Okay, sorry, this will be here. Then this is the CRP binding site, promoter. Then we have the operator. Okay, so repressor is already bound with the allolactose. Okay, so it will not be able to bind the operator. First thing, then the RNA polymerase is here, and here the due to high levels of CMP, the CAP protein is also bound to the RNAP. Okay, due to high levels of CMP. So this is active. Okay, now what are the conditions that might be present here? Now, allolactose is present, that means lactose is present in the medium, and CMP levels are high, that means glucose is absent in the medium. Okay, so this is the case. If glucose would be present, then CMP levels would be low because of the metabolism. And due to this low CMP levels, the cap protein would not bind to the RNA polymerase. So the glucose is not there, and lactose is present because the repressor is already bound to the allolactose pair. So glucose and lactose are present? No. Glucose is absent. Glucose is present, lactose is absent? No. Because if lactose would not be present, then allolactose would not bind to the repressor. Both are present? No. Then glucose is absent and lactose is present. So yes, the correct answer is this one. Okay. So yes, the correct answer is option 4. Moving on to the next question. Two E. coli cultures A and B are taken. Culture A was earlier grown in the presence of optimum concentration of the gratuitous inducer IPTG. Both the cultures are now used to inoculate the fresh medium containing suboptimal concentration of the gratuitous inducer. It was observed that the culture B was unable to utilize lactose, whereas culture A did so efficiently. The reason behind this is so, first thing, IPTG is, as we have discussed in the applications for the lacoperon. So, what it does is it's basically isopropyl beta D 1 thiogalactoside. Okay, so it's a isomer of the lactose or allolactose only. Okay, and it is also called as a gratuitous inducer because it induces the lacoperon but is not metabolized by the cell. Okay, so what we have is we have two cultures A and B E. coli. Culture A was earlier grown in the presence of optimum concentration of the IPTG. So here we have IPTG ka concentration and it was grown first in this. Both the cultures are now used to inoculate fresh medium containing suboptimal concentration of the inducer IPTG. So now from this, it has been given another culture in the fresh medium that have IPTG in it, that has lower concentration of IPTG as compared to this. Okay, and here it was not grown in IPTG. Now, it was observed that culture B was unable to utilize lactose. Here, lactose was not utilized. Okay, whereas in culture A did so efficiently. So, what would be the reason for this? So, we'll see the options first. Pre-treatment with IPTG has resulted in a mutation as a result of which lac operon is constitutively expressed. Okay. IPTG has made the cell membrane more porous to small molecules, and so lactose is taken up more efficiently by A as compared to B. Okay. In culture A, lactose permease was induced to a high level during pre-treatment with IPTG, which allowed the preferential uptake of lactose. Okay. In culture A, IPTG activated a receptor which bound lactose more efficiently and thereby triggering a signal. Okay. So what will happen? IPTG can induce the lacoperon without metabolizing. Okay. So what would have happened in culture A when IPTG was added? So the lacoperon will be activated, leads to production of all the three enzymes: beta galactosidase, lactose permease, and the transacetyls. Okay, so all these three were present. Then what will happen? Lactose permease is also already present in the system. So what would be the case when lactose permease would be there? It would have formed the transporter to selectively transport lactose inside the cell very efficiently. Now, in B case, what would have happened? The lac permease level would be very low or null, leads to the very low uptake of lactose. Okay, so the bacteria could not utilize the lactose very efficiently as compared to A. Okay, so the correct answer is option C, because in culture A, when IPTG was there, then it would have induced high levels of lactose permease. This is increased in the cell, and during which allowed the preferential uptake of the lactose. That's why A was able to grow in the culture in the lactose containing medium as compared to B. Okay, so the correct answer is option C. Okay, now moving on to the next question. E. coli was grown in three different experimental conditions. In one case, okay, so the first case, the grown medium containing glucose as carbon source. So E. coli was grown in glucose. In the second case, there was glucose and galactose, and in the third case, it was infected by a phage. Okay, now match the curve shown below with the treatment. Okay, so now when the bacteria would be grown in the, we'll see with the first case. Okay, so here are the number of cells when it will be grown in single carbon source. Okay, so it will follow a sigmoid curve growth. Okay, so what will happen? It will have a log phase followed by a exponential phase, and then it would go to the saturation and decline phase. So this is a typical sigmoid curve that will be taken up by most of the bacteria when grown in single carbon source. Okay. Then when we have the two carbon sources, that is glucose and galactose, or any other carbon source, then it will have a dioxic curve. Okay, so that will be something different as compared to the sigmoid curve code. So what will happen? It will have a step in between like this. Okay, so this will be the characteristics of the two carbon source. So this was the first and this was the second. Now when infected by the phage, what will happen? The number of cells will first increase, then during lysis the number of cells will decrease. Okay, so this would be typically like this kind of code. Okay, so this would be the 3-1. So in the question, it was Q, this was P, and this was R. Okay, so P would be glucose plus galactose, Q would be glucose only, and R would be with phage, infected with phage. So let's see the options. So P is grown in glucose, Q is grown in both glucose and galactose, and R is infected with the phage. The R1 is correct, but P1 should be with glucose and galactose. So first option is wrong. Then we have P grown in glucose and galactose. Okay. Q in glucose, fine. And R is infected with the phage. Yes, this is the correct option. So the correct answer is option B. The correct answer is option B. Okay. Now moving on to the next question. You have created a fusion between the tryptophan operon, which encodes the enzyme for tryptophan biosynthesis under the regulatory control of the lac operon. Lac operator. Under which of the following conditions will tryptophan synthase be induced in the strain that carries the chimeric operator fused operons? So we have done is we have the promoter, then we have lac operator followed by the tryptophan synthesis genes. Okay. Now when there will be a lac operator, then this would be controlled by the lac repressor. So we have to check when tryptophan synthase gene would be expressed in the bacteria. So when both lactose and glucose are absent, so lac repressor will only work. Sorry, will bind to the lactose. Lac operator when lactose or allolactose is not present here. But when lactose or allolactose will be present, then it will bind to the repressor and tryptophan synthesis will be expressed. So this condition where lactose must be present, and the second case is with the glucose. Okay, because glucose inhibits the lac operon with the other pathway. Okay, due to catabolite repression, so the glucose must not be present here. Okay, so the first option is wrong because lactose is needed. Then we have lactose and glucose both present. So in that case, the tryptophan synthesis will be the genes will not be that much expressed. Okay. Then we have only lactose absent and then glucose is present. So in that case, the repressor will not work and the synthesis of these particular genes will not take place. Then we have only when lactose is present and glucose is absent. So yes, the correct option is option D, because the lactose will bind to lac repressor that will free the lac operator so that RNA polymerase can synthesize these genes. And the if glucose is absent, so that will increase the CMP cap complex and that will bind to the RNA polymerase, thus increasing the amount of transcription of these particular genes. Okay, so the correct answer is option D. Okay, now moving on to the next question. A recombinant protein is to be expressed under the control of lac operator and promoter, promoter and operator in a strain of E. coli having the genotype lac I plus and CRP plus. Okay, so this is the recombinant protein, and this is the I, that is inhibitor or the lac repressor of uh, that is the repressor of the lac operon. Even in the absence of inducer IPTG, low levels of expression of the recombinant were seen. That was leaky expression. Okay. Which one of the following should be done to minimize such leaky expression? Okay. So what the thing is, we have the CRP plus protein and then we have the LACI. Now, this CRP plus is in the under the control of the operator. Okay, now this will go and bind to this particular operator that will not allow the transcription of this particular protein. Okay, now what will happen if some glucose and lactose both are present, so there will be some leaky expression that has been mentioned in the question. So we have to decrease this leaky expression. How can we do that? So in that case, lactose or any inducer must be not present, first thing. And how we can minimize? We can increase the concentration of glucose. Okay, because of the catabolite repression, it will not allow the from like operon system to be activated. So here, when glucose's concentration would be high, so that would decrease the expression of the recombinant protein. Okay, so addition of lactose to the medium, no, we do not need this because we have to minimize the leaky expression. Then we have removal of all glucose medium, all glucose from the medium, no, this also cannot be done. Then we have addition of excess glucose to the medium, yes, and allolactose also is an inducer, so this is also we need to not to be done. So the correct option is option C, because here we need to stop the lacoperon. So the correct answer is option C. Okay, now moving on to the next question. In context to lacoperon, if two bacterial strains P1 and P2, so here we have P1, P2. P1 has a genotype OC, I plus, Z minus, and P2 has the genotype OC, I minus, and Z plus respectively, were used to produce mero-diploid daughter strain D. So these were fused and a mero-diploid strain was produced, that is D. Which one of the following statement correctly predicts the expression of Z gene, that is beta galactosidase activity, in all the three strains? So, okay. So the genotype of D would be OC, I plus, Z minus, and O plus, I minus, Z plus. Okay, because this is the mero-diploid daughter strain. Okay, now considering the phenotype of, so we have to detect the expression profile of the beta galactosidase gene. Okay, so in P1 there is Z minus, so no beta galactosidase activity. This is the first scenario. Okay, talking about beta 2, sorry, P2, we have Z plus and O plus and I minus. Okay, so although in P1 we have constitutive operator, thus the inhibitor or the repressor cannot bind to the operator, but beta galactosidase is absent, so there will be no activity. Now, in this case, we have the Z plus, that is beta galactosidase present. The operator is normal and I is mutated such that it will not be able to bind to the operator. Okay, so in that case, what will happen? we'll see constitutive beta galactosidase activity. Okay. Now in the third strain, that is D. So we have the genotype OC, I plus, Z minus, and O plus, I minus, Z plus. Okay. So we'll draw that figure again. In this we have OC, I plus, Z minus, and this we have O plus, I minus, and Z plus. Okay. So here, from here there is no activity, so we'll not refer to this, but this would be problematic. So what will happen? Here we have the repressor. Okay, repressor is trans binding, so it can bind to any operator who have the similar sequence. So it will bind to this particular operator, and it will be inducible in nature. Okay, here the operator is constitutive, so it will not be able to bind, the repressor will not be able to bind with this operator. But here we have the operator wild type, that is normal. So here the repressor will bind and will only be relieved when the lactose or allolactose is present. Okay. So in this case, the beta galactosidase activity will be inducible. Okay. So P1 will have no expression, P2 will have constitutive expression, and D will have inducible expression. So let's see the options. Now we have P1 as no expression, and in so we'll see in the elimination mode now. P1 would have no expression. So in ABC there is no expression, but in D there is inducible expression. So this option is eliminated. Now we have P2 that shows the constitutive expression. Okay, so P2 will have constitutive expression, but it will not have inducible expression. So C is also eliminated now. Now D would have the inducible expression. So the correct option is option A. This is the correct option. So the correct answer is option A only. So this is wrong. This must be option A. Okay. Now moving on to the next question. A miro-diploid strain of E. coli with the genotype. This is F positive, OC, Z negative, Y positive, and then we have O plus, Z positive, and Y positive. So this depicts the F plasmid was constructed. The activity of beta galactosidase enzyme was measured in this strain upon following treatments. So first we'll see how will the Z plus will act here. Okay. So now we have the constitutive operator and here the wild type operator. Here the constitutive operator will have no use because the beta galactosidase gene is not here. And here we have the inducible operator. So the beta galactosidase activity will be seen only when lactose or any inducer will be present. Okay, so this whole system would be inducible now. Okay, now we'll see in different scenarios how will the graph look. So first is no induction, no activity. Okay, then we have B, induction with n moles of IPTG. Okay, so yes, it will have beta galactosidase activity. Then we have n moles of lactose, same level of activity. Okay, So suppose this is X activity, so this is will be also X activity. Okay, then we have D, that is n moles of lactose in the presence of n moles of glucose. Okay, so here there will be catabolite repression and there will be minimal beta galactosidase activity. Okay, so here will be less beta galactosidase activity. Okay, so now if we draw the graph, this in the y-axis we have beta galactosidase activity, and on the x-axis we have treatment. So in the first case where there was no induction, there will be zero activity. So this is A. Then we have B. So in B we have, suppose, this much activity. Then in C also we'll have the same activity. Okay. Then in D we'll have less activity. Okay. Rather, in case of lactose, when there is a, so here there is one trick. Okay, IPTG is not metabolized by the cell. So the whole IPTG can be used for induction of the lactoperon. But lactose is also metabolized by the cell. So here we'll see a bit less activity as compared to the the IPTG induced treatment. Okay, so I'll change it. So here the activity would be this much, that is a bit less than this, but in the last case we have less activity. Okay, so based on this the correct graph would be option B. Okay, so option B would be the correct graph. Option 2. So uh, here there is one trick that I completely forgot. So what will happen in the A part, because we have seen that when the lactose is not present, there is small amount of beta galactosidase activity present due to leaky expression. Okay, so here in the A part, it will not be completely zero. It will be similar activity as compared to the D1, because here the glucose will apply the catabolite repression. Okay, so the correct answer will be not B but C. C would be the correct answer, because here there will be some activity upon we do not have add any inducer, and the similar activity will be present when there will be glucose present in the medium. Okay, so the correct answer will be option 3. Now, moving on to the next question. A gene encoding for protein X was cloned in the expression vector under the T7 RNA polymerase promoter and lac operator. Cells were induced by the addition of 1 millimolar IPTG at 37 degrees Celsius for 6 hours. Cells were lysed and fractionated into insoluble bodies, and cell-free supernatant by centrifugation was collected. Protein X is present in the insoluble bodies. Which one of the following strategies would you use to express protein X in the soluble fraction, that is cell-free supernatant? So what will happen? So first we'll understand what is the PET system. Okay, so this particular system, where T7 RNA polymerase is used under the control of the lac operator. So this system is called as PET expression system. Okay, and this is the basic idea about the PET expression system. Okay, first we'll understand this, then we'll move on to the question. So what will happen? We have the lac operator and promoter, and then we have the T7 polymerase gene. Okay, when we add the IPTG, this E. coli polymerase will act upon itself because the lac repressor cannot bind here, and then T7 polymerase will be produced. Okay, this T7 polymerase is like now can act upon the different this T7 polymerase now can act upon the, because it has higher efficiency for the T7 promoter. So now what will happen, this T7 promoter will now express the target gene in higher concentrations. So this is considered to be a good expression vector in bacteria. So here we need the IPTG induction. So this was the basic idea about the pet vector system. Now we'll get back to the question. So now we've induced these cells with 1 millimolar IPTG, 37 degrees Celsius and 1 hour. And then cells were lysed and fractionated into insoluble bodies and cell-free supernatant by centrifugation. So here we collected the cells and then we divided the cells using centrifugation using cell-free supernatant and the insoluble bodies. Okay. Now, which one of the following strategies would you use to express protein X in the soluble fraction? So, okay, so the protein is now observed in the insoluble fractions, that means as an aggregate. Okay, why aggregate? Because high amount of protein is formed. This was the case. Okay, so now what happens? High amount of protein is formed, and then what we have to do is we have to either increase induction time, increase temperature, or increase concentration. These three only uh, ways we have to increase the expression of the protein in the soluble bodies. Okay, so now we can increase the duration of the induction. Okay, lower temperature, no, lower temperature will not work here. We can increase the concentration or increase the temperature. Now here the click is the T7 RNA polymerase promoter basically works at higher temperatures. Okay, so it will be around 50 degrees Celsius. So here if we have to have the protein in the soluble fraction, okay, in the cell-free supernatant, then we must increase the temperature to 50 degrees Celsius. Okay, that's why the correct answer would be option 4. We have to grow the cells at higher temperature after induction with 1 millimolar IPTG. That's why it will, if we grow the cells in lower temperature, then it would be going into the insoluble fraction. Okay, now moving on to the next question. The DNA segment was cloned into a active site region of LAC-Z gene, and the recombinant plasmid introduced into the LAC-Z minus strain of E. coli and plated on a medium containing X-gap. The colony showed blue color. Which of the following statements is correct? Okay, so here they are basically showing the blue-white selection method. So what happens in blue-white selection method? When suppose this is the LAC-Z gene that codes for beta galactosidase. By any chance, we introduced a DNA segment into this, so that what will happen? It is interrupted and no active protein will be formed. Okay. So now what will happen? Here we'll have two scenarios where the gene is normal and where we have an extra segment in the gene. So here normal beta galactosidase will be present, and here no beta galactosides will be present. So after adding the substrate X gal, this will show. So after adding X gal, it will show blue color, and here it will not show blue color, it will be white in color. Okay, so now we have the XZ negative strain of E. coli where we have introduced this particular gene containing the DNA segment. Now we have added the X gal containing X gal in the medium, and the colony showed blue color. So blue color would be visible because beta galactosidase is normal. Okay. So nature of the cloned DNA segment need not be special, as cloning of any DNA in laxet will result in disruption of its reading frame and production of blue color on the X gal plate. No, because the DNA segment will disrupt the protein sequence of the beta galactosidase. So it will not produce the functional enzyme, hence the X gal cannot be converted to blue color. The cloned DNA segment could be a group 1 intron whose removal from the precursor laxet transcript in E. coli results in the production of mature laxet mRNA, which can then produce active laxet protein. Okay, so this can be the case because, as you all know, group 1 protein, group 1 intron is basically a ribozyme. Okay, so it can self cleave itself. So if there would be a group 1 intron in the system, it would have removed from the system while expression, and the original gene is like reconstituted following the production of the beta galactosides. The cloned sequence is likely to be lac sequence, which is naturally a part of the lac operon in E. coli. No, because then also the beta galactosidase sequence would not be restored. The cloned sequence is likely to be an anti-terminator sequence, which allows full-length transcription of the laxet gene. No, the anti-terminator only like stops the like does not allow the transcription to be stopped. Okay, so this do not have anything related to the transcription. So the correct option is option number 2. The likely correct option is option number 2. Okay, so this marks the end of our PYQ session for the prokaryotic transcription. Now we'll be starting with the eukaryotic transcription for now. Now, as we already like discussed before, the basic idea about the transcription unit is that there is a structural gene that codes for a protein. Before that, we have a promoter, that is the binding site for RNA polymerase basically, and the terminator that basically stops the transcription. So this is the basic idea about the transcription unit of any organism. Now, what we have is in prokaryotes we have normal promoter, then we have the structural gene, and we have various structural genes arranged in the form of an operon followed by a terminator sequence. But in eukaryotes it's an entirely different thing. The basic sequence is same, but the gene sequence and the number of parts present is different. Okay, so we have the in eukaryotic gene structure, we have the upstream regulatory sequences that basically consist of the promoters, that these are proximal control elements and the promoter. Okay, so this is the major promoter that is being recognized, and then we have certain elements like enhancers or silencers. These are called as distal control elements that controls the binding of RNA polymerase and various transcription factors on this particular proximal control element sites. Okay, so this is one thing. The second thing is downstream regulatory sequence. These are present towards the 3' end. Okay, so this will contain the terminator sites and the enhancer or silencer, that are distill control elements. Okay, so this is the second regulatory sequence. Now going back to the structural gene that codes for a protein. So in case of eukaryotes, we have a specific uh, structure such that there are two kinds of sequences present. One is the exon and one is the intron. So in basic terms, the exon is the coding part and intron is the non-coding part. So exon basically, these number of exons are combined with each other to form a complete mature mRNA chain that leads to protein, the final protein. And in this process, the introns that are non-coding, so they do not code for any protein, so or yes, so that are removed in the process. So they are removed via RNA splicing, that we'll see in the RNA processing wala part in the next week. So this is one thing, and in eukaryotes there is only a single RNA or single gene associated with one promoter and terminator. That's why the RNA of the eukaryotes is called as monocistronic RNA, because one promoter is associated with one gene. Sorry. So the one promoter is associated with one gene. In case of prokaryotes that we talked about, one promoter is associated with many genes. Okay, so that is why it is polycistronic. It is called polycistronic, and here the promoter is called as monocistronic. Okay, so these were different structures that are being present in the eukaryotic genes. Now, we'll see different promoters and how they function. So here we have the promoters. So in case of the eukaryotic promoters, they are weak promoters. Okay. But in case of prokaryotic promoters, they are really strong promoters. That is why they do not require much of the activators. But there are certain weak promoters also, like lack promoter, that is a weak promoter. That's why it requires a activator. And most of the eukaryotic promoters are weak promoters. That is why they have a specific site for enhancer silencers that increases the transcription activity. Now we have three different RNA polymerases, so three different promoters. So we have three kinds of RNA polymerases that function in the nuclear cells. Okay, so that is why we have three different promoters in that system. Then we have what will happen in case of the prokaryotes. As you remember, the sigma subunit of the RNA polymerase recognizes the -10 and -35 sequences of the promoter, and then it will bind and initiate the transcription process. But in case of eukaryotes, what happens? First, the transcription factor binds to the promoter. This facilitates the entry of the RNA polymerase binding. Okay, so here what happens? The transcription factors, there are different transcription factors for different genes. So each transcription factor first recognizes whether there is a need to transcribe that particular gene or not. Okay, so that is why first the transcription factor binds to these particular elements, and their different transcription factors binds. That facilitates the entry of the RNA polymerase. Okay, so this is the thing. And then here in the eukaryotic system, the promoters are generally present, like the whole sequence of promoter is present either downstream or upstream. So it can be downstream also in case of eukaryotes, but in prokaryotes it's already upstream of the transcription start site. So considering this as the transcription start site, okay, so this will be plus 1, right? So the promoter can extend from here till here. That's what we mean. Okay, so it can be extended both upstream and downstream of the transcription start site. So these are some of the properties of the eukaryotic promoters, and then we'll see in detail, a bit detail, about the different types of promoters. So here we have three different RNA polymerases: RNA pol 1, RNA pol 2, and RNA pol 3. Okay, now talking about the RNA polymerase 1 promoter, so it has basically two components. One is the core promoter and one is the UCE, that is upstream control element. So the core promoter ranges from -40 to +20 region. So the transcription start site is basically in between this core promoter. Okay, and here the transcription bubble also starts. So this would be a bit AT rich sequence. Okay, now we have the upstream control element, that is a GC rich region. Okay, and it lies between -180 to -100 region, and it is a regulatory sequence. So here only the activators binds, that increases the activity of the RNA polymerase. Okay, so if this sequence is removed or mutated, then the transcription would follow normally, but the level or rate of transcription would go down. That's why it's only the binding site of the activators. Okay, so this was the thing with the RNA polymerase 1 promoter. Then we'll move on to the RNA polymerase 2 promoter. So RNA polymerase 2 promoter is the well studied promoter in the eukaryotic system, and it has various regions. It has it has the core promoter. Starting with the core promoter, so core promoter has two regions. One is the Tata box and one is the INR region. Okay, so the Tata box is basically a -30 position. Like in prokaryotes we have -10, so here we have the -30 position, and it is also called as Hogness box. Okay, and the INR sequence is basically ranges from -2 to +5. So the transcription start site is in between this particular sequence, and it is also partially located upstream of the transcription start site and partially downstream of the transcription start site. Okay, and it has a specific sequence of two pyrimidine nucleotides followed by an A. So generally the start site starts with the A. Okay. Then we have another nucleotide that can be any either of the four. Then we have two pyrimidine residues. So this was the consensus sequence found by the scientist. So this is a specific sequence for the INR sequence. Okay. Now talking about the regulatory sequence, we have two box, CAAT cat box and GC box. So the cat box is present at the -70 position and GC box at the -90 position. So here the GC box has a sequence of 3GCGG. So this is the sequence of the GC box, and it is highly conserved. And these two sequence work in orientation independent manner. That means if the CAT box is first as compared to the GC box, then also it will work in the same way. Okay, so this is the thing with the regulatory sequence, and here the all the activator and transcription factors bind. So on the Tata box, it facilitates the binding of the transcription factors that recruit the RNA polymerase. Okay, So these are the stuff. There are certain more sequences, like we have three more sequences. First is the one that lies here. This is a BRE. So what uh, then here after the INR sequence, we have two sequences. First is the MTE and the DPE. So BRE is basically TF2B binding site. Okay, so the name is TF2B recognition element. Then we have the MTE or the motif 10 element. Then we have the DPE or the downstream promoter element. Okay, so these sequences also help in regulating the transcription using the RNA polymerase 2. Now moving on to the RNA polymerase 3 promoter. So it is divided into two types, that is upstream located or downstream located. So here it is based upon the TSS, that is transcription start site. Okay, so it can be upstream of the transcription start site or downstream of the transcription start site. So in upstream case, it is a type 3 RNA polymerase 3 promoter which encodes U6 snRNA. Okay, and in the downstream case, it's type 1 and type 2 RNA polymerase 3 promoter. So type 1 forms 5s rRNA and type 2 forms tRNA. Okay, so in the type 3 case, which is upstream located, so we have a normal data box and -30 position. Then we have two more sequences, PSC and OCT. So PSC is present between -60 and -40, and OCT is present in between -240 and -200 sequences. And OCT stands for octameric sequence or the DSC, distal sequence element. Okay, and PSC stands for proximal sequence element. So these are basically regulatory sequences that helps in binding of different activators, or that increases the activity of the RNA polymerase. And then we have the type 1, we have box A and box C. Okay, that ranges from +50 to +70 for box A, and box C is +80 to +90. These are downstream of the TSS. Okay, then we have the type 2 RNA polymerase 3 promoter, in which we have box A and box B. So box A is between +10 to +30, and box B is between +50 to +70. So this was the thing about the RNA polymerase 3 promoter. Then moving on to the different types of RNA polymerases. So the nuclear RNA polymerase first. Then we have majorly three RNA polymerases: one, two, and three. So we'll see this is based on the function, and then we'll see the sensitivity of these particular RNA polymerases at certain more information. So now the RNA polymerase 1 basically forms the large RNA rRNA precursor. Okay, and these basically forms the 28S, 18S, and 5.8S RNA. So the RNA polymerase 1 basically helps in the formation of rRNA. Okay, all the major rRNA present inside the ribosome are formed by the RNA polymerase 1. And this rRNA large precursor is 45S. The RNA polymerase 2 basically forms the mRNA. Okay, the precursor for the mRNA is hnRNA, that is the precursor form of mRNA, where it contains the introns. Now the spliced version is called the mature mRNA, and the snRNA is small nuclear RNAs. So these forms the mRNA and snRNA. Now we have the third RNA polymerase that basically forms the tRNA precursor, U6SNRNA, 7SL RNA precursor, and 7SK RNA precursor. So tRNA, so third RNA polymerase, that is RNA polymerase 3, basically forms the tRNA. Okay, and U6SNRNA involved in the spliceosome complex, and 7SL and 7SK are small RNAs. Okay, involved in various regulatory processes. So this was the thing. So this type of questions also are asked in the competitive examination. Thus the information based questions. So RNA polymerase 1 encodes all type of rRNA except for the 5s rRNA that is encoded by the RNA polymerase 3. Okay. The RNA polymerase 2 encodes mRNA and snRNAs. RNA polymerase 3 encodes tRNA, 5s rRNA, and various small RNAs. Okay, so these were the basic functions of the RNA polymerase 1, 2, and 3. In plants, we have two more RNA polymerases that were identified, RNA polymerase 4 and RNA polymerase 5. So, RNA polymerase 4 basically helps in siRNA production in plants. Okay, and 5 is helps in regulation of siRNA. So these were the two more RNA polymerases added in the list, but these are only present in plants as of now. They have not been identified in in animals. Okay, moving on. So these RNA polymerases has a sensitivity to drug that is alpha amnatin. Okay, so alpha amnatin was isolated from alpha amnatin was isolated from a mold basically, or a mushroom that is named as Amanita phalloides. This was the name of the mushroom. This is a poisonous mushroom that basically yields a toxin alpha amnatin. Okay, so these three polymerases 1, 2, and 3 have different sensitivity towards alpha amnatin. So seeing this figure, we can see the activity of different polymerases with alpha amnatin. So polymerase 1, as you can see, has no effect on the activity when applied with the alpha amnatin. So in this case, it's resistant to alpha amnatin. Talking about RNA polymerase 2, so it is highly sensitive to alpha amnatin. Then polymerase 3 has less sensitivity as compared to polymerase 2, and this is basically species specific sensitivity. Okay, so this is the case. This is the resistant one. Polymerase 1 is resistant to alpha amnatin. Polymerase 2 is highly sensitive, and polymerase 3 has less sensitivity as compared to polymerase 2, and it depends upon the species to species. So RNA polymerase 2 has 1,000 times more sensitivity of alpha amnatin as compared to RNA polymerase 3. Okay, so based on this experiment, it was identified the different sensitivity nature of alpha-amnatin to different RNA polymerases. Then we have the structure. So here we'll compare the structure of RNA polymerase that is present in prokaryotes and in eukaryotes. So one thing, in prokaryotes, the RNA polymerase that is present, that is single type. Okay, so all functions, all RNA produced by that particular polymerase is uh, like are transcribed using same polymerase. And in eukaryotes, we have three different polymerases, one, two, and three, for different functions, okay, because of the complexity of the organism. Then in the prokaryotic polymerase we have beta prime beta, alpha 1 alpha 2, omega, and the sigma. Okay, now we'll compare different subunits with the RNA polymerases. So the basic nature, because it has been seen that we have evolved from the prokaryotes only, so the basic function of the subunits remain the same, but the number of subunits would have been increased. Okay, so as you can see, the first and second subunit of all the three polymerases is similar as the beta and beta prime. So these are the polymerase subunits. Okay, then we have the alpha like subunits. So these basically help in assembly as well as binding to the activator sequences. So UPE elements in the prokaryotes. So this is the similar function of this particular thing. Then we have omega like subunits that simply maintains the structure of the RNA polymerase, core RNA polymerase. Then we have some common subunits based on the species, because these are present in eukaryotes, so these are some similar structural subunits. Then we have the additional enzyme specific subunits. So in total, these are the more units present in the RNA polymerases. Okay, so the main purpose of showing this was to show that different subunits of the prokaryotic RNA polymerase also has, like eukaryotes also has similar performing function subunits also. The number might vary. Okay, now moving on to the RNA polymerase 2. This is well characterized RNA polymerase in eukaryotic system, and why it is important because of its special nature of activity. Okay, so it has a C terminal domain that has, this is called as a C terminal tail. Okay, so this particular domain of this RNA polymerase is the largest domain of this enzyme, which has an extension at C terminal tail. It contains a heptapeptide. Okay, so heptapeptide that has a sequence of tyrosine, serine, proline, threonine, serine, proline, serine. So it's a serine threonine rich sequence that is basically phosphorylated during the RNA polymerase reaction. Okay, so the series or the position of phosphorylation shows what type of function needs to be done next. So for example, if the process of initiation needs to be changed with the elongation, so the phosphorylation pattern changes. Okay, so in that sense it changes with the process. Then what phosphorylates these particular residues is the kinase activity of the another transcription factor, that is TF2H. So it has a kinase activity that basically phosphorylates the tail time to time so that it can change the process. And why to phosphorylate? Because this phosphorylation attracts different proteins that are important for the different processes, because here a lot of transcription factors need to be used for elongation, for initiation. So this phosphorylation is important for attracting these particular proteins so that the process needs to be like accelerated, and this helps in recruitment of different transcription factors, this phosphorylation. Okay, so this is a peculiar feature of the RNA polymerase 2, and this also recruits all the enzymes involved in capping, splicing, and tailing. Okay, that is why there is a hypothesis that states that only the RNA polymerase 2 formed RNAs, okay, are only processed in capping, splicing, and tailing. Okay, so this has been hypothesis for long time, and no proper evidence has been found yet. So this was the thing because of the tail present, and it recruits all the proteins involved in this particular processes. So this tail also helps in RNA processing, in short. Okay, now there is also in eukaryotic system we have extra chromosomal DNA also present in the form of the organelles. So there are two organelles, mitochondria and chloroplasts, that have their own plasmid DNA. So there might be some RNA polymerase present in those organelles also. So that is also called as organelle RNA polymerase. So in case of mitochondria, we have nuclear. So in case of the organelle, we have two kinds of RNA polymerase. Some subunits or whole is encoded by the nucleus, and some encoded by the organelle itself. Okay. So in case of mitochondria, we have a totally nuclear encoded RNA polymerase, and so that means all the subunits will be encoded by the nucleus. Okay, and it has both core enzyme and sigma factor that are derived from the nuclear genome. Okay, and then we have the chloroplast. So it has both nuclear encoded and chloroplast encoded. So the nuclear encoded part, where whole core and the sigma factor are encoded from the nuclear genome, and the chloroplast encoded, where the core component is from the chloroplast, but sigma factor is from the nuclear endpoint. Okay, and the major thing is because these two, mitochondria and the chloroplast, have been thought to be evolved from the prokaryotic system. So this RNA polymerase is basically similar to the eubacterial RNA polymerase. Okay, that is why the sigma factor and the core enzyme has been named. So this was the thing about the organelle RNA polymerase and different RNA polymerase present in the eukaryotic system. Before that, we have studied about the promoters and present in the eukaryotic system. So that's all for today's class. We'll see you in the next lecture of the RNA biology lectures. Thank you for your attention.